Hello. Last time we were talking about Gregor Mendel and him being the father of genetics, and we were talking about a P generation who is true breeding, crossing with another of a P generation, which is true breeding and creating an F1. So let's continue where we left off. So we're going to talk about probability today, and we're going to continue with Mendel's genetics. So probability is the likelihood an event will occur. It's measured as a ratio of the desired outcome over all possible outcomes. Now that's a mouthful. Let's actually look at a problem and see how this works. So you know that if a coin is tossed into the air, what is the probability that coin will land on heads? Now you know that probability will be 1/2, but let's break this down. So we know the favorable outcomes. The favorable outcome is heads. We know that all the possible outcomes are heads or tails, and we can see those possibilities here. We know there's only one way to get heads, so that's what we have for the numerator or for the first number. And for our denominator or second number, we know we have two possibilities. That's why if we toss a coin in the air, the probability that it'll land on heads is 1/2 or 1/2. So what is the probability of a couple, they want to have children, what is the probability of this couple's first child being a girl? Well, there's one desired outcome, and there are two possibilities. One could be a girl, the other could be a boy. So again, this is also 1/2. How about if this being a boy? Well, the same thing is true. We have 1/2 possibilities. Let's try this. What is the possibility, or what is the probability of dice, or a single dice, being rolled and getting an even number? Now on a dice there's 1 through 6, there's numbers 1 through 6. So 2, 4, and 6 are the numbers that we're looking for, and we can get 1 through 6. So we have 3/6 possibilities of rolling an even number. When we take that number and reduce it, we again get 1/2. So we have a 1/2 chance of rolling a single die and getting an even number. So let's see how this applies to genetics. Okay. So we can use this information to make a prediction in biology, specifically in the field of genetics. So let's take a look at what this looks like. So we've investigated a little bit about segregation. So during gamete formation, alleles for each gene segregate from each other so that each gamete carries only one allele for that gene. So let's look at how this works. We've talked about in a previous video, if you cross a true breeding tall plant, and that true breeding tall plant, since it's true breeding and tall is dominant, the genotype for that plant would be capital T, capital T. A true breeding short plant would be lowercase t, because short is recessive, and true breeding means the two letters are the same. And Mendel figured out that the results are all tall plants, and you can see that here in the F1 generation. So he took a true breeding tall and crossed it with a true breeding short plant and got all tall plants. And he was a little perplexed by this. He actually did this with all 7 traits that he studied, but we're going to follow this through with just a height characteristic. So what he figured out was segregation had occurred. So to find out what had happened to the recessive allele, Mendel allowed all 7 of the F1 hybrids, there's a term that'll be helpful. He allowed these F1 hybrids to self-pollinate. In other words, he allowed them to pollinate with themselves. So the offspring of an F1 generation is called the F2 generation. Bet you can figure out what the offspring of an F2 is called. Hopefully you said an F3. If you did, you are correct. The F2 offspring of Mendel's experiment are shown here. So you can see he got 3 tall out of 4 plants. He got 1 short out of every 4 plants. Now, that doesn't mean that he got 4 plants. He could have gotten hundreds and hundreds and hundreds, but 75 out of every 100 would have been tall, and 25 out of every 100, roughly, would have been short, or pretty close to those numbers. Notice the ratio in the F1 generation. This is very important. When you have a hybrid crossed with a hybrid, you always get a 3-to-1 ratio. You get 3 of the dominant trait, or in this case tall, to 1 of the recessive traits, or in this case short. The reappearance of the recessive trait in the F2 generation indicates that at some point the allele for shortness had actually separated from the allele for tallness. So let's take a look at how this happens. This is going back to meiosis. So let's explain this. So how did the segregation or separation or segregation of the alleles occur? So there's something called the principle of segregation. Mendel suggested that the allele for tallness and shortness in the F1 plant must have segregated or must have separated. This is called the principle of segregation. They must have separated from each other during the formation of sex cells or gametes. So let's take a look at what we're talking about here. So in the F1 generation, we said the F1 were hybrid. Now, that has an implication. What that means is their genotype was capital T, lowercase t. And this one plant that we're talking about is going to be both the mother and the father. So this is the F1 generation. Now, in order to figure out how or what kind of offspring these plants can produce, first we need to make the gametes. Because in order for an organism to reproduce, they have to have sex cells, and those sex cells are gametes. So when we look at this first plant, we look at what kind of T can this plant give to its offspring. And we notice it can give a capital T. Now that capital T will go into one gamete, so I'm going to circle this. Anytime I draw a circle around something like this, it's representing a gamete. But they can also create a gamete with a lowercase t. So that's for this parent. For this other parent, you find, well, it's the same plant, so it can produce the same alleles. So this is one plant crossing with itself. This would work with this plant crossing with itself. It would also work with this plant crossing with another hybrid plant. Once we have the gametes, what we can do is we can then combine these gametes with each other. So if we do something called FOIL, this should sound familiar, possibly from math. If we take the first of each gamete and combine them, we would have an offspring that has a genotype of capital T, capital T. If we take the O, which is outer, that's the outer letter in each case, we would have a capital T, lowercase t. So let's put a comma between that so we know that's 2 different individuals and it's the genotype. I is inside, so we're going to take the inside letter from each of these, and we have a capital T, lowercase t. In that case, you can see these 2 are the same. And then we have last. We're going to take the last letter from each one. And now we know we have every possible combination if we crossed these 2 plants. Well, you can see that we have this plant, which is homozygous dominant. Oops. And we have, so that is this one plant. We have these 2 plants that are heterozygous. And we have this one plant that is homozygous recessive. Now, you should notice that these three plants, these are all tall. So we have 3 tall to 1 short. Now, we'll always get this 3-to-1 ratio when we have a hybrid crossed with a hybrid. Doesn't matter what kind of organism this is. So you'll always get this pattern. So these are how gametes are formed. So when we take a look at this, we've got, you can see over here, we've got the capital T, lowercase t. You can see how we've got our 4 different types of gametes. These different types of gametes, so these are the 2 from this parent, these are the 2 from this parent. Then when you take those gametes and you cross them, you can see how you get these 4 genotypes, just like we've worked out. So what we can do is, instead of drawing all that information, we can take these F1 plants, we can figure out the gametes, which we figured out are capital T, lowercase t for this individual, and we've got capital T in one gamete and lowercase t for the other gamete, so these letters separate from each other. It's just like when you have a homologous pair, the homologs separate, and then each of those replicated chromosomes will separate. What's doing the separating are these alleles. So instead of crossing and drawing all these lines that we've got here, what we can do is we can create something called a Punnett square. Now in this case, our Punnett square is going to be a 2 by 2 square, because we have 2 gametes for this individual, we have 2 gametes for this individual. So what we're going to do is we're going to take the gametes from one of the individuals, and we're going to put each one of these circled information on top of each column. Then we're going to take this second individual, excuse me, and we're going to put their gametes down the side, and we're going to fill this in just like a multiplication table. And this will show us all of our possible offspring. So the probability of getting tall, we can see here that these individuals will be tall. The probability of tall is a 3-to-1. The probability of short, the probability of tall is 3 out of 4. The probability of short is 1 out of 4. The ratio, let's write that over here, the ratio of the genotype would be 3-to-1. So our probability is the number of desired over the total number of individuals. Again, it's the total number desired over the number of individuals. The ratio of the genotypes includes how many of each genotype. So we always write the dominant first and the recessive second. Okay. So let's try a monohybrid cross problem. That's exactly what we just did. A monohybrid cross is a one-trait cross. In other words, we're examining only one trait when we cross 2 individuals. So we're going to cross a true breeding pea plant with green pods. True breeding, that means both letters for this individual are going to be the same. With a true breeding for yellow, both alleles for this yellow plant are going to be the same as well. The way we know that is because we have the terms true breeding. Now here's our clue. Yellow pods are recessive. So the yellow's recessive. That means the green is, you're right, it's dominant. So we're going to use a capital G for green. And in this case, since this green pod plant is true breeding, we need 2 capital G's. We also have a true breeding pea plant that has yellow. So since yellow is recessive to green, we have 2 lowercase g's as the genotype for that individual. So our parent generation is capital G crossed with lowercase g. Now you'll see that this Punnett square is going to be much more simple because our gametes are fewer. The only type of gamete this individual can make is a capital G. The only type of gamete this individual can make is a lowercase g. So since we have one gamete and one gamete, we have a 1 by 1 Punnett square. So we take all of the gametes from this individual, which are 1, we put those gametes at the top of each column, and we take all of the gametes from this individual, which are 1, and we put them on the rows, and we fill this in just like a multiplication table. So we can see that all of the offspring, all of the F1 offspring are going to be, they're going to have a G, capital G, lowercase g genotype, and they're going to be what color? Hopefully you said green. That would be the phenotype. So let's try another one. What is the probability of the offspring having green pods? It would be 100%, as you can see right there. They would all be green. So in certain plants, yellow fruit is dominant to white fruit. So we're going to use a capital Y since yellow is dominant. And I'm going to write my lowercase y like this. Now Y's are a little bit of a challenge. You've got to make sure they're distinctly different in the way you write them. A heterozygous, heterozygous, that means they have 1 of each letter. Now, we notice this is in parentheses. We would not need this clarification with yellow fruit because if they're heterozygous, we know they have 1 of each letter. If they have 1 of each letter, that means they have the dominant allele. That means that dominant allele says that this plant will have yellow fruit. Now, let's move on. So it's crossed with a plant with white fruit. White fruit, white is indicated by a lowercase y. Well, in order for this to be white, it's got to have 2 lowercase y's, because if we had an uppercase y, this plant would be yellow. It would have yellow fruit. So we now have this as one parent and this as one parent. So let's take our parental generation. We have our capital Y, lowercase y. We have our lowercase y. And it says, what are the possible genotypes? So we want genotype and phenotypes of the offspring. So again, our gametes we need to produce. Okay, here we've got a capital Y and a lowercase y. And over here, we only have a lowercase y. So we can see here that we've got a 2 by 1 Punnett square. So let's create this off to the side here. So we can do a 1 by 2 or we can do a 2 by 1. So we can draw this either this way or this way. Either one of these will be correct. So let's use this first one. We have a lowercase y for this individual, and over here we've got the 2, so we have a capital Y and a lowercase y. So this offspring would be capital Y, lowercase y. This offspring would be lowercase y. So you can see that we have one individual that is, what color is this? That is correct. This is yellow. And we have one individual that is white. So we have a 1-to-1 for our phenotype. And you can see that we have yellow and white. So it asks what are the genotypic and phenotypic possibilities. Our genotypic, we can see here inside the Punnett square, that would be capital Y, lowercase y, and lowercase y, lowercase y. And over here I'm going to put that this is a 1-to-1 ratio. And then we know that, so that's our genotypic. And our phenotypic, we have yellow. I'm going to name that first because it's dominant. And we have white. Name that second because it's recessive. And I'm going to put here that it's a 1-to-1 ratio. So these two pieces of information will be our answer for the possible geno and phenotypes, because you can see here's our genotypes, here's our phenotypes, and it also answers this. What is the ratio of the genotypes and phenotypes? You can see here's our ratio of our genotypes, our dominant, and then our recessive. Here's the ratio of our phenotypes. And that's a monohybrid cross problem. There's also something called a test cross. You can have an individual that demonstrates a dominant characteristic. And looking at this organism, you don't know if they are homozygous dominant or if they are heterozygous. These two individuals would look exactly the same. So we can perform something called a test cross. A test cross is a cross between an organism that exhibits the dominant trait, just like these two, with an organism that exhibits the recessive trait. So we would cross this unknown with this type of individual because we know this demonstrates the recessive trait, therefore it must be homozygous recessive. And we're talking about this for the same gene. And we can do this in order to determine the genotype of the dominant trait individual or the dominant trait organism. So we're trying to find out if this individual has this genotype or if it has this genotype. So let's take a look at how this works. So we have a purple flowering plant and it's crossed with a white flowering plant. Now that's interesting. We have purple and we have white. We don't know anything about this at this point. So in this species of plant, purple is dominant to white. So if purple is dominant, we're going to use the letter P. Now the letter P can be somewhat dangerous to use. If you have if you have a capital P, you should write it this way. If you have a lowercase p, you may want to put a line underneath it. As a matter of fact, with a capital, you might want to as well, unless you have lowercase p's around somewhere. So I know I have a capital P plant, but I don't know what the second letter is because it could be capital, it could be lowercase. I know I have a white plant. Since it's white, I know I have two lowercase p's. There's our test cross. So in this species of plants, purple is dominant to white. The results, we have a 35 purple ratio to 7 white. This looks very close to a 3-to-1 ratio. So let's take a look at this. If we have, so let's take a homozygous dominant individual. Let's try, let's assume this individual is capital P, and we're going to cross this individual with our known lowercase p. The only kind of offspring these individuals can produce is purple. The reason why is because this individual can only produce capital P gametes, And this individual can only produce lowercase p or white gametes. And when we perform the Punnett square, we can see these are all purple. So could this be the genotype of our unknown plant? No. Here's why. We got white flowers when we crossed them. So let's check out this one. Could it be a capital P, lowercase p that we crossed with our lowercase p, lowercase p? Well, we've got capital P gamete and lowercase p gamete. We've got it crossed with a lowercase p gamete. Draw a line there. So for this Punnett square, I'd have a capital P and a lowercase p for this parent, and a lowercase p for this parent. And when I fill in my Punnett square, we can see this way we have the probability of getting white offspring and purple offspring. So this parent must be, this purple flower must be this genotype. So it says, what is the genotype of the original purple flowered plant? So we know it is capital P, lowercase p. That is our answer. So let's try one more. In yellow pea plants, yellow seed color are dominant to green seed color, and they've already given us the letters that we're going to use. A pea plant with yellow seeds, so we know this individual, we know one individual has capital Y, and we don't know what the second letter is because they're yellow, and they could be capital Y, or they could be capital Y, lowercase y. We don't know for sure. The resulting offspring have about equal numbers of yellow and green seeds. So we know that we cross this yellow seed plant with a green seed plant. A green seed plant must be lowercase, lowercase. So, and just like before, if we get both yellow and green seed plants, before we calculate anything, we know that this individual has to have this unknown second allele, has to be lowercase y. It's the only way to get green and yellow offspring. Because green offspring are lowercase y, lowercase y. This y had to come from that parent because this y came from that parent. So this, the genotype of the parents would have to be capital Y, lowercase y, and we know that the green one had to be lowercase y, because otherwise you couldn't get, otherwise it would be yellow if it had a capital Y. So in another cross, let me let you try this. And if you want, I can tell you what the answer to this is, what the answer is to this if you come join me in Zoom. And that's it.